Okay. Morning, everyone. I'm Neil Nolan. I'm a third-year resident in the Mass General Brigham Harvard Neurology program, and I'm excited to introduce our MGH Grand Round speaker today, Dr. Cheng-Hua Gu. So Dr. Gu received her PhD at Cornell Medical School, followed by postdoctoral training at Johns Hopkins. She came to Harvard Medical School as faculty, where she's now a professor of neurobiology. She's a pioneer in the field of neurovascular biology and has made large contributions to our understanding of the molecular mechanisms behind the blood-brain barrier, as well as neurovascular coupling, the process by which neural activity increases local blood flow to meet local brain metabolic energy demands. Her most recent work on this active role of endothelial cells in neurovascular coupling was published in Nature earlier this year. Speaking on behalf of Doug Wilcox, one of my co-residents who is working with Dr. Gu. Dr. Gu and Doug will be starting a project soon addressing how the blood-brain barrier serves as an immune signaling platform between inflammatory stimuli in the systemic circulation and the brain parenchyma. The immune response of the blood-brain barrier has important implications for a wide variety of neurologic conditions, including infections of the brain, endothelial dysfunction in SARS-CoV-2, and a variety of neuroinflammatory and neurodegenerative disorders. In recognition of her groundbreaking work, Dr. Gu has won numerous awards, including an Allen Institute professorship and the NIH Director's Pioneer Award for highly innovative research. She is currently an HHMI faculty scholar. Thank you, Cheng-Hua, for speaking at Grand Rounds today. And if there's any extra time at the end of the session, I'm happy to facilitate questions from the audience, which can be posed either through the chat box or the hand raise function. Thank you.
Great. Thanks, Neil. Thanks for the kind introduction. And also, I hope Doug will feel better soon. And I hope everyone in your family stay safe at this difficult time. And thanks for coming to my seminar. Okay, so, oops. Okay, great. So the title of my talk is the neurovascular interaction of the brain. I just want to remind you, maybe it's less necessary for this audience. So this is actually a human brain vascular cast, just to give you a sense how densely vascularized our brain is. Indeed, every neuron is less than 15 micron away from a capillary, and also from energy conception point of view, brain is a very expensive organ. Even though it's only occupied 2% of a body weight, it consumes 20% of energy at rest. Like right now, you're listening to my talk, and with very limited storage ability. Finally, also, we know neurons are extremely sensitive to their extracellular environment. So facing those challenges, luckily, our brain also evolved to have a unique way of supply the blood to the brain, which is very different from the periphery. There's two key unique features. So one is to meet the moment-to-moment energy demand. Neuroactivation increases local blood flow called a neurovascular coupling. Secondly, this chemical milieu must be tightly controlled, and this is by the blood-brain barrier. So my lab, as Neil mentioned, in the past couple of years, we've been really focused on trying to understand the fundamental molecular cellular mechanisms underlying these two unique physiologies of neurovascular interactions in the brain. So today I'm going to share with you a story of each, and just to give you a sense where we are and talk about what's ongoing work too. So I'll start with the blood-brain barrier. First, I'd like to acknowledge the people who have done the work. So this is initiated by a very talented former postdoc, Eyal Ben-Zivi, with help with another former postdoc. Both of them have their own lab now. And then the more recent work done by two very talented graduate students from the PIN program, also former graduate students, they already moved on. So as a neuroscientist, we all really appreciate the importance of the blood-brain barrier. It's really a gatekeeper to the brain. It's basically control the in and out of what the molecular substance in and out of the brain, and keep the ion nutrient homeostasis, and also protect from the toxins and pathogens with very limited entry of the immune cells. So here, just to it's a cartoon version of the blood-brain barrier. Is basically constituted in a single layer of the endothelial cells, and here's the astrocyte. Try to illustrating the end feet hugging the blood vessels, and there's another cell type called parasite, which you couldn't see here, but I'm going to mention that too. So no doubt have a very tight regulated blood-brain barrier is very important for the regular brain function, but at the same time is also the biggest obstacle for treating any CNS diseases. So all the stuff I listed here, right now, the biggest obstacles is very limited drugs can actually pass the barrier. So perhaps in this case, we really wish we could transiently open the barrier to let this drug go in. On the other side, this is more emerging research suggests that barrier breakdown precedes the neurodegeneration. So in this case, we probably wish we could tighten the barrier to delay the disease and progression. So I think our job is to try to understand the fundamental mechanisms underlying the blood-brain barrier, understand how it's built and maintained, and that will enable us to gain tools and knowledge to enable the therapeutics. So just a little bit history, the concept of the barrier actually arrived in the early 1900s by this famous experiment done by Paul Ehrlich and his students. Basically, they inject the water-soluble, in this case, blue dye, to the rat and then tail vein, and then the rat run around for some time. They just simply dissect all the organs, and was something very surprising and astonishing is that except the brain and the spinal cord, everything else turned blue. So there's many interpretations of this experiment, but one of them was perhaps this indicate there's a physical barrier between the blood and the brain. So this concept to actually realization, demonstration of where the barrier actually is took another 60 years until the seminal work done Tom Rees and Morris Kronowski in the 1970s. So that time, the state of art technology is the EM. So what they did is they inject the horseradish peroxidase, which can react with DAB, become electron dense, visualized under the EM to the mouse tail vein, and then same thing, cut the brain. And here you can see under the EM, here's the lumen, and here you see the tracer stop very sharply at the tight junctions between two neighboring endothelial cells. So the reason is a seminal experiment because this is for the first time actually demonstrating the barriers localized in the endothelial cells. Prior to this, I think many neuroscientists speculate this should be in the end feet of the astrocyte. Even nowadays, people still feel like this is a property for the astrocyte. So, but this is very definitive. They also did a reverse experiment. If you inject tracers in the brain parenchyma, the same thing. They stop very tightly, very sharply at the tight junction between the endothelial cells. But I also want to point out in the same paper, in the same animal, if you look at the periphery blood vessels, so here is the lumen, it's quite opposite. You see the tracer actually goes through the tight junctions. So this is a stark contrast. And in addition, they also show a lot of vesicle containing tracers in the periphery endothelial cells, but they don't really see this in here. And you can see the tracers even stay in the basement membrane. So because of this result, the major conclusion is that one of the major contributor for the restricted nature of the blood-brain barrier is this very called specialized tight junctions. Even today, we really don't fully understand what do you mean specialized in the molecular cellular term, but functionally it's day and night difference, right? So this is tightly like no water soluble molecule can go through. So I just remember when we first started this line of work, this work really inspired me a lot. I look at this picture staring for many, many times and just astonished by this. And so the simple question we asked back then is that, what are the molecular determinants underlying those unique properties, give rise to those unique properties? So to start to probe this question, what we did, Ayal did, is that we isolated endothelial cells from the brain cortex and from the lung, and we performed the back then just microarray, and then very clearly so each dots represent one of the gene transcription, and most of the genes are shared, right, between these two populations, because they, at the end of the day, they're both endothelial cells. However, we're particularly interested in this, the red dotted genes. So show here, right, they're very enriched in the cortex, but almost minimal expression in the lung. So there's about 200 candidate genes and encoded transcription factors, transmembrane proteins, transporters, regular transporters. But for today, I'm just going to share with you one of the genes called MFC2A. It's a multi-transmembrane protein. We studied, this is the first one we studied because it's about 80-fold enrichment during development, 800-fold enrichment in adult compared to the cortex and the lung. So I'm going to share with you what we learned from study this MFC2A. So first of all, the pure reason, because a lot of people ask me, you have 300 genes, why you pick this one, right? So the pure reason is that we actually first saw in the Allen Brain Atlas that if you look at mRNA expression, it's only in the brain, the spinal cord. And if you zoom in, it looks like vessels, not neurons. And we later developed an antibody. You can see absence of the mutant mice and stain the protein also very clearly stain the blood vessels. And here we don't have to go through details. Basically, just saying it's only expressed in the endothelial cells, but not even neighboring parasite. Okay, so what is MFC2A? The official name is Major Facilitator Superfamily Domain Containing 2A. It's mouthful, it doesn't really tell you that much. So it's a 12 transmembrane protein. Back then, we didn't really know too much about the function, so we decided to look at the knockout mice and see whether there's anything to do with the blood-brain barrier. So here's the bread and butter type of functional assay. So we inject, in this case, it's a red tracer. It's about 10 kD Dalton. You inject to tail vein, and then this is the brain slice. And you can see in the normal situation, the wild-type mice, tracer completely confined in the blood vessels. However, in the MSU2A mutant mice, you see the tracer leak out of the blood vessels, stain the brain parenchyma. Surprisingly, this is actually not due to tight junction defect. Here, basically, we did exactly the same experiment Morris and Tom did. The tracer, you can see in the mutant and the wild type case, both stop very sharply at the tight junction, indicating the tight junction is functional normally. However, when we look at transcytosis, this is wild type case, as Morris and Tom saw before. In the wild type case, the tracers completely stay in the lumen. You don't see anything in the endothelial cells. However, in the mutant, basically, this is a reminiscence to what they saw in the periphery, right? So basically, you see the tracer containing vesicles in the, oh God, why does this keep doing that? In the endothelial cells. So this is very exciting because basically mutate this one gene, you kind of converted CNS endothelial cells to the periphery, at least from the transcytosis point of view, right? And it's really telling us that seems like it's not like the endothelial cells in the brain, they don't have the transcytosis mechanism. Rather, they're being actively inhibited. So the idea, with a cartoon version, previously we think we all know, major contribution to this restricted nature of blood-brain barrier is somehow the brain endothelial cells have very specialized tight junctions, preventing this paracellular trafficking. So here our data indicate that unlike periphery endothelial cells or many other cell types, substance can also take in by vesicles, trafficking, released to the other side. And this mechanism is being actively inhibited in the brain endothelial cells. So we think this is one of the new mechanisms underlying the blood-brain barrier. So to further develop this concept, so we asked the following 3 questions. One is, does this apply to other blood CNS barrier, right now in the spinal cord? And also, can transcytosis be dynamically modulated? And finally, what exactly the mechanism? So since it's all published, I'm just going to use one slide to give you a take-home message here, and then to discuss this a little bit more because it's more in depth. Okay, so to make a long story short, we look at during development, turn out to be that when the vessels first enter the CNS, they're leaky, just like the periphery, and but surprisingly, even that time when it's leaky, the specialized tight junctions actually is already formed. You just see a lot of vesicles, and then gradually those vesicles are trimmed away, and then only when it's totally suppressed, the barrier become functional. So this tells us that the rate of transcytosis actually is a determining factor for the functional barrier formation. So we actually demonstrated in the brain retina barrier. So it just demonstrating this mechanism is quite widely applied to other barriers. So what is the mechanism? So first of all, we look at MFC2A protein expression. This immuno-EM, so basically you can see they're not expressed in the tight junctions, expressed on the luminal side of the membrane, sometimes associated with vesicles here, right? So here you see. So then the first question we want to know is what type of vesicles are involved, right? So here just a summary of, cell biologists discovered a variety of type of vesicles and with different molecular markers. To make a long story short, we found in the blood-brain barrier in the MFC2A case is the Cavioli vesicles are specifically suppressed. So Cavioli vesicles are the small plasma membrane invagination, and the Cavioli-1 protein is the obligated component to form those vesicles. So here's the evidence. In the MFC2A mutant mice, you can see the immuno-EM of CAV1. You can see those CAV1 decorated the Cavioli vesicles. But to definitively demonstrate this, we did mouse genetics. So here's some evidence. So we're demonstrating MFC2A suppressed transcytosis by specifically blocking the Cavioli formation. So here I show you already MFC2A mutant mice have a lot of vesicles. I color in here for you. Oh God, why do they keep? Okay, so however, when we cross MFC2A mutant mice with Cavioli-1 mutant mice, Cavioli-1 without Cavioli-1, there's no Cavioli vesicles, right? You can see here, and this is completely rescued this phenotype. You see very minimal Cavioli vesicles there. So mirror this result. I don't know how to mirror this result. The leakage phenotype also completely rescued here. So this means that the normal function of MFC2A is to actively suppress the Cavioli formation, in turn maintain the blood-brain barrier integrity. So we wanted to know how this happened, right? So there's many possibilities, right? It could be physically interact with Cavioli pathway, could suppress the Cavioli pathway by some adapters, also could control the plasma membrane tension, therefore affect the Cavioli formation. So as we were just scratching our head, thinking where to begin, our companion paper actually published in the same issue by David Silver's group in Singapore, what they found is that MS2A is a transporter for the essential omega-3 fatty acid DHA. And later, two groups also demonstrating the natural mutation happening in human and then required for omega-3 fatty acid transporter. So, oh my God. Okay, so here with a cartoon version, what it's saying is that the MFC2A, this is a green cartoon, basically it's a transporter for the phospholipid like DHA. So you can flip that from the outer membrane to the inner membrane. And then we show it's involved in the transcytosis. So whether these two have anything to do with each other, I think is quite important, even though on the surface sounds like not. So we decided to do a experiment to definitively testing whether these two functions related or not. So to do that, we took advantage of that. The MSC2A is a syn port. In order to transport a lipid, it actually first to have to bind to the sodium. And this site is conserved among different species. So what we did is basically we mutate this key residue D96 to alanine. So it's a simple point mutation, knock-in mice. And here shows even though just one amino acid change is completely abolish its lipid transport function. So this is lipidomics done by Clara Klisch in the Broad Institute. Basically look at both the knock-in mice compared to its control versus the full knockout with the control. You can see that completely recapitulate each other. So it means even though it's a 1 point mutation, it's completely abolished lipid transport function. So what happened to this mouse? Well, basically it's a completely recapitulate the full knockout phenotype, have increased transcytosis. Here's the quantification, and here you can see the tracer containing vesicles in the knock-in mice, and the junctions completely normal. And here's the leakage, right? So basically means the lipid transport function is actually critical for blood-brain barrier. So we know it's just a single point mutation. So this actually made us to think maybe it's actually direct role of the lipid involved in the BBB. So basically the idea is that maybe MFC2A impedes the Cavioli vesicle formation simply by altering the luminal plasma membrane lipid composition, so prevent the vesicle budding. So turn out to be this is not a very crazy idea. If you look at the literature in the cell biology, a lot of this all from in vitro work by Kai Simon and others, showing that actually in the plasma membrane, the Cavioli domain usually are enriched in the very organized, like classroom type of lipid, and devoid from those very fluid, and unsaturated DHA type of lipid. So so you can imagine when you enrich the lipid bilayer with this species, then you actually could displace Cavioli 1 and cholesterol. So we think this maybe will happen at the BBB. To show this, what we did is we asked Clara Klisch to do the lipid lipidomics experiment for us again. So again, this looks familiar. This looks familiar as the transcriptome analysis I showed you before compared to the lung and the brain capillary, but here is actually lipidomics. And you can see, you can see the lipids also, most of them are also shared. However, among this differentiated level of lipids, the red dots is a DHA containing phospholipid. So you can see majority of them actually is indeed the DHA containing phospholipid. So, so here, it's a very important piece of data. I just want to say here because as I mentioned in the both knockout mice and the human mutation for the MFC2A single mutant, you have microcephaly, you also have a blood-brain barrier defect. So, however, I want to point out that when we do the double mutant mice, they rescued the blood-brain barrier, but failed to rescue microcephaly. The reason I think this is important because it's demonstrating that the blood-brain barrier opening is not the secondary of microcephaly or some neuronal deficit. So just to put everything together, so we think at least that this is one of the first mechanism underlying how transcytosis inhibition can contribute to the blood-brain barrier integrity. So in the case of the lung or the periphery endothelial cells, they can naturally form Cavioli vesicles. So as a result, they if you do a snapshot for the EM, you see plenty of those vesicles, and they can do their job, transcytose. However, in the brain endothelial cells, because of the unique expression in MFC2A, which can flip the DHA type of a phospholipid from outer membrane to inner membrane, changing the fluidity of the plasma membrane. So as a result, it's an environment naturally inhibit Cavioli type of vesicle formation. So therefore, if you do a EM snapshot, very rarely you actually see those kind of vesicles. So obviously, this is just the tip of the iceberg. So currently we're looking for other key molecules, right? Clearly we have 278 candidates. MFC is just the first one we're studying. So we have a pipeline right now go along the list. And right now we have like 14 or so already being tested, and we have like handful already give us the leakage phenotype. So they seems revealing all kinds of underlying mechanisms. Hopefully in the future I can share some of this with you. And in addition, we also start to go besides endothelial cells, because we know endothelial cell is not intrinsically determining the blood-brain barrier function. It's actively need to be maintained by the astrocyte and parasite. For example, if you take the piece of tissue out from or you dissociate the endothelial cells from the brain, just a couple of hours, molecules like MFC2A just disappeared, okay? So you really need this unique brain environment to maintain the barrier. So we wanted to know what are the factors coming from astrocyte and parasite induce this. And finally, as Neil mentioned, Doug is working on with us is how do the endothelial cells transmit the peripheral immune signals to the brain. It's sitting right in between them, and what kind of role is those brain endothelial cells maintained. Okay, so I think for the second part, I'm going to switch gear to share with you a most recent story about neurovascular coupling. So this is done by Brian, a former grad student, and Vince, also a former postdoc. They both just left the lab in January. Okay, so I just want to remind you, during behavior, various brain regions are recruited to perform specific tasks, right? So the resulting neuroactivity rapidly increase the local blood flow. So this phenomenon is called neurovascular coupling. So this is a dynamic interaction is very important for day-to-day function of the brain, so that it can meet the moment-to-moment changes in the regional brain energy demand. In addition, this is also the basis for the functional brain imaging in humans, like fMRI, and also more recently, there's an emerging set of studies indicating that its impairment also contributes to neurodegeneration. Despite the importance, our understanding of neurovascular coupling, fundamental mechanisms still in a very still not very clear, partially because this level of resolution, that's not enough to study the temporal or spatial resolutions, not give enough to understand the mechanism. So my lab have been doing for the past few years is to develop a new paradigm to study this. So basically, we use two-photon microscope and cranial window to look at the mouse brain in a weak mouse. So here I'm going to show you a movie. We're looking at the barrel cortex because when you stimulate the whisker, you can see the response right away. We can synchronize these two events. So here, if I click here, you see this movie. You can see whisker stimulation, and here's a GCAM indicating the increase of the neural activity, and followed by, so here's the labeled single arterial blood vessels, and you can see the wall dilate and then come back. In addition, we also have the capability to image the capillary blood flow. So together with mouse genetics, we start to use this combination set of techniques to explore new ideas of neurovascular coupling. So here, I just want to remind you that the blood vessel in the brain, you're coming from the pier, large vessels come down very quickly, become very elaborate capillaries. So the neurovascular coupling depends heavily on the smooth muscle cells because they are the ones having the capability, have the contractile capability. But I also want to remind you, they're only cover the large vessels, but not the majority, 90% of the capillaries. So in the field, so far, most of the study have demonstrated that neurovascular coupling, the general view is that it's basically neuroactivity from neurons and maybe astrocyte involved can directly sense vasodilatory cues to the smooth muscle cells, and that's the reason why you see the neurovascular coupling. However, as I point out, many neurons are very far away from this, and this in vivo process from starting of neuroactivity to vasodilation is hundreds of thousands millisecond, hundreds of milliseconds, okay, so very fast. So by diffusion of those cues, it's probably not sufficient enough. So at least for the neurons far away from the smooth muscle cells. So I'm going to share with you a story. Hopefully in the end, I'm going to convince you that endothelial cells actually can play an active role in this process. So the reason we get into this actually very serendipitously because of our barrier work. So if you see here, it's a slice from the brain slice, and Claudin 5 labels all the vessels, and hydrazide is the label the large arterial vessels. And you can see if you do MFC2A staining, one thing is very interesting that the capillaries are positive, but the larger arterial vessels, they're negative. So that means MSC2A only expressed in the capillaries, but not in the arterial vessels, right? So then the question is, I just spent the whole half hour convince you that the major function of the MSC2A is to inhibit Cavioli formation. So, and we know naturally in the wild type mice, the capillaries have very low level of Cavioli. So is this means if you look at arterial endothelial cells, they should have a lot of Cavioli? So indeed, that's a case. So here, the capillary, you can see very minimal. Arterial, we see a lot of vesicles, right? And we know there are Cavioli vesicles because if you look at in the mutant, they all disappear, right? Cavioli 1 mutant. Oh God, and here it's probably very, very clear. You can see so many of those vesicles, flat shape vesicles. And I also want to point out that not only in the endothelial cells, also in the smooth muscle cells, you can see here. So this is really interesting, right? So here's a quantification. You can clearly see the day and night difference between the capillary and the arterial endothelial cells. So the question is then, what is the function of high level of Cavioli in the AECs? Because arterial endothelial cells is a site of vasodilation, so we figure since we have the setup, why not we look at the Cavioli knockout mice, right? So again, it's the same movie, just a different mouse, but you probably already noticed that although you still have the neuroactivity increase, but the blood vessel wall seems just stuck there, tried to expand but couldn't. So if you do a quantification, I think here just to show the movies again, this is a group result, and basically you see about 50% reduction of the diameter changes. However, the baseline is no change. The basal level of the vessel diameter is no change. And here, just to demonstrating, we're able to do 2P actually quite deep. Here's like 300 micron, and this thing is no difference. Okay, so it's not really propagation problem. It's basically in all the arterial endothelial cells that we see this deficit of neurovascular coupling. So here is the capillary blood flow I mentioned. You inject tracer in the blood vessels, so the red blood cell will appear dark, everything else will fluorescence. So based on the shadow, you can calculate the speed of the, the speed of the flow. And you can see here in the mutant mice is dramatically slower than this. It's also 50%. Here's a quantification, also 50%, and there's no phenotype in the heterozygous mice. So both the vasodilation and capillary blood flow is 50% reduction. So the first thing we thought will be, because this is a general knockout mice, and we know Cavioli also expressed in the smooth muscle cells, so maybe there's something wrong with smooth muscle cells. So we look at smooth muscle cells and turn out to, here just to show you, they're wrapping around the arterioles and seems normal, and you quantify them, everything's fine. And here's the functional assay. So basically, if we're directly giving an NO donor through the PIA into the live awake mouse, and you can see the vasodilation to the same degree in the mutant and the wild type, means the smooth muscle cells still have the capability to the normal contractility. So all of this point to the possibility that maybe arterial endothelial cells cell autonomously, the cavioli in the arterial endothelial cells cell autonomously required for neurovascular coupling. So to do this, luckily, we have conditional now, cavioli 1 conditional now mice, and also BMX is the arterial endothelial cell specific driver. So basically, we cross the crate to the flox mice, and you can see here, if you do a CAV1 staining, as I said, both arterial endothelial cells and the surrounding smooth muscle cells all have CAV1, right? But in the arterial specific mutant, the middle part, right, arterial endothelial cell CAV1 disappeared, and the surrounding smooth muscle cell CAV1 still there. Okay, so what happened to this mouse? So before that, you can also predict if that's the case, then you should selectively reduce cavioli in the arterial endothelial cells, but no change in the smooth muscle cells. That's exactly what we saw, right? So here you see much less cavioli in the arterial endothelial cells, and you see plenty in the smooth muscle cells. And here's the quantification. Okay, basically means we successfully ablate the cavioli only in the arterial endothelial cells. So what happened when you reduce the arterial endothelial cell cavioli vesicles? Well, it's basically completely recapitulate the full knockout phenotype. You see about 50% reduction, both blood flow and vasodilation. In contrast, if you do the smooth muscle knockout, specific knockout, you have no phenotype. Okay, there's still normal. The match 11 with the same mutant mice is both blood flow and there's no difference from the wild type. So this is, I think, definitively demonstrating that in the case of the arterial endothelial cells, it has very low expression of MSH2A. As a result, have a high level of Cavioli, and this is required for neurovascular coupling. And in the first part of the talk, it's just in a different type of endothelial cells, right? The capillary endothelial cells, they have a high level of MSH2A. As a result, have a low level of Cavioli, and this low level of Cavioli is very important to keep the blood-brain barrier integrity. So then we want to push this one step further. So we say, what if we artificially increase the MSH2A in the arterial endothelial cells? Will that also reduce Cavioli and then have neurovascular coupling defect? So it's a basically different way to manipulate Cavioli to test this hypothesis. So we made a mouse, which is Cre-dependent MFC2A expression mouse. Basically, we can cross that with BMX Cre artificially only in the arterial endothelial cells, increase the expression. So here you can see in the wild type case, I showed you before, SMA, this is the arterial endothelial cells, they're negative for MFC2A. However, in our new mouse, when you cross with the BMX Cre, you can see those arterial endothelial cells now express abundantly MFC2A. And importantly, you also see the arterial endothelial cells have much, much less, almost minimal level of arterial Cavioli vesicles, right? So here's the quantification. So basically, and there's no change in the smooth muscle cells. So again, what happened to this this mouse? And same thing. So basically, ectopic expression of MS2A in the CNS arterial endothelial cells attenuate the neurovascular coupling, both dilation. So here's a dilation. So you can see about 50% reduction. There's no difference in the baseline level. So basically, this result together point out to a new model that in addition to this direct signaling from neurons to smooth muscle cells, perhaps there's another pathway exist, which is especially in light of a recent work by Mark Nelson's group showing capillary endothelial cells can sense the increased neural activity. So perhaps the signal can propagate it, we think most likely from gap junctions to the arterial endothelial cells, and then, or the data I'm showing you right now indicating that arterial endothelial cells can talk to the smooth muscle cells in a Cavioli-mediated dependent manner. So currently, obviously this is a very new idea, require many labs, many work to demonstrate this. But I think this is pointing to the fact that through this whole process, endothelial cells can play an active role. They're not with thought as before, they were just passively being dilated. Okay, so then go back to the story, one thing we still haven't really understand is how do arterial endothelial cells utilize the Cavioli to mediate neurovascular coupling, right? So Cavioli have a very rich literature, plays so many functions. So one of them is interact physically with endothelial NO synthesis, ENOS, and then also can serve as a membrane reservoir during the mechanical stretch. Transcytosis I already mentioned, also propagation carried by connexins, and also cluster iron channel and receptors. Among all of this, we think, NO is a prime vasodilator, so perhaps this is worth to test, right? So we decided to do a double mutant mice. So the idea is that if they're in the same pathway, then you should see a similar effect, right? If they're in a different pathway, you should see additive effect. So here is the result. If you see a single mutant of either ENOS or CAV1, you get 50 or 40% reduction. However, when you have mutant mice, which is the purple one here, basically it's like flattened. You almost completely abolished neurovascular coupling. This is like not what we expected at all, but it's a very, very clear result. And here's the capillary blood flow. So this is indicating the pathway we discovered actually is different from the ENOS pathway. They're running in parallel, but both of them together knock out, I would say over 95% of the neurovascular coupling. So indicating is a very important pathway. To make a long story short, we also excluded all the other possibilities. We think the most likely function of a Cavioli 1 here is probably cluster ion channels and receptors. So currently we have some candidate and we're working on it. So here, just to summarize what we found, obviously again it's just the tip of the iceberg, and currently we try to understand how the neural signal propagates through the EC, the endothelial cells, to the smooth muscle cells. But equally important, we also want to know how this propagations get stopped, right? Because it's only local blood flow response to increased neural activity, not the entire brain get vasodilation. And finally, now we have a beautiful, very clean double mutant genetic model, this double mutant mice. So we wanted to know what are the consequences of the impaired neurovascular coupling for the brain function. So I think that's the time. It's good. I think I mentioned the people done the work, and here's the Clara Klisch did the lipid domic analysis, and I didn't show the data from Adam's lab. This is more ongoing work, and here's we get the mice. I guess here's a funding. So here's my lab. Right before the COVID lockdown, we were out celebrating the paper. So Doug is not here yet. Okay, so thank you. So I'm going to take questions.
Okay, thank you very much. So if anyone has any questions, you can list them in the chat or the hand raise function.
So I should stop sharing, I guess. Yeah.
Let's see. Okay, looks like we have a question. Okay.
Hi, this is Merritt. First of all, I just want to thank you for an amazing talk, amazing body of work. I was just curious on your thoughts about any how this might change with aging, or kind of where to go for maybe therapies around some of this great, elegant work.
Yeah, so it's a great question. I did not mention, because my lab is now focused on the aging type of research, but for example, Tony Westcory in Stanford recently showed that, in the aged mice, they saw the reduction of the MFC2A. And we also know various labs also have showing that in the aging mice, the barrier also become leaky. So I think it's all go to the same direction. This maybe contribute to some degree because it's decreased the level. And then from therapeutics point of view, also the other direction, right? As I mentioned, there's urgent need for opening the barrier in certain degree. So this is, as an inhibitor, it's a perfect candidate, because if you can repress the inhibitor, basically you can transiently open the barrier. So my lab actually actively develop such reagent. We're trying to develop a functional blocking antibody to inhibit MFC2A function, transiently open the barrier.
Thank you very much.
Mm-hmm.
Okay, thank you. And so we have a few questions in the chat. First from Dr. Lanoila, asking what other pathways of blood-brain barrier regulation are you exploring other than lipid mechanics?
Yeah, so I think the ongoing work seems like showing not necessarily Cavioli is the only type of vesicle. We also find some potentially other type of vesicles, so they may not involve in lipid mechanism. I think that's one example. We also find things affect tight junction, modified tight junctions function also. So this is still early. We're investigating the underlying mechanism.
Okay, next question from Patricia Staley. Do you think there are genetic disorders of lipid metabolism, for example, adrenoleukodystrophy, in which the MSFD2A fails to inhibit caviole? Is that known?
No, I don't know. Well, what is adrenal? I don't know this disease. Disorder. Yeah, what is this? Adrenoleukodystrophy.
I could invite Patricia to speak if just one moment here.
Yeah. Adrenoleukodystrophy.
Okay, I'm having some trouble with that, but it's a.
I see. Genetic disorder.
Maybe we can move on to the another question and come back in a moment.
Okay.
So I see Peter Rosenberger asks, you're suggesting that brain tissue is unique in its ability to regulate the blood supply to meet metabolic demand. Similar mechanisms must exist in other tissues. Is this true in muscle, gut, or other tissues? Is that known?
Okay, can you say it again? I missed the first part.
Oh, sure. Peter Rosenberger is suggesting and asking if similar mechanisms to regulate blood supply to meet metabolic demand exist in other tissues like gut or muscle.
Yeah, that's actually a really good question because which I think is certainly not in a lot of places because you don't have this really local, very tightly local, very calculating way of reserve your energy, to local response, right? But one place we think the most similar may be the muscle. There are some mechanisms maybe share, including the, gap junction involvement, electrical conductance. But for many other places, it is different.
Okay, thank you.
Mm-hmm.
And Dr. Mukherjee Shabani is asking if there are known polymorphisms for MFSD2A and caveolin, and how do knockout models handle the acute insults like infection or TBI?
Yeah, so because we notice in our own hand, seems like they're more prone to the infection because we do cranial window. Usually, based on what we do, the wild type mice, they're much, they can last for like several weeks and they're no problem. The few cases we use MS2A, just you get infection. That's my impression.
Okay.
Yeah, I don't know about traumatic brain injury. Yeah.
And then Martin Zhang posted a question in the chat asking in general about whether there have been studies on exercise or sleep and the changes those might have on the molecular changes you saw in your studies.
I think people started to work on this. I think there's a paper in Drosophila, just talk about blood-brain barrier during, Drosophila circadian rhythm. Definitely difference between day and night. So I speculated maybe this is true for the higher level organism too. Is the area need more research on.
Okay, thank you. So I think we have time maybe for a couple more questions. So Florence Lai is asking if there is a connection between a leaky blood-brain barrier and high cholesterol.
Yeah, so I mean, because I think when you do lipidomics, there's a lot of, even on your mutant, you see the increase of cholesterol, right? So we actually trying to do diet type of experiment is always in combination of a lower DHA, high cholesterol. So the kind of you couldn't tell that's a correlation or consequences of that or not. So I think there is type of correlation there. Yeah, so I think I don't have a definitive evidence, but I definitely, yeah.
Thank you. So Rachel Bennett asked a question. I'm sure people were wondering these CAV1 ENOS double knockouts had what you described as a major loss of neurovascular coupling in it. We were wondering if they exhibited a phenotype as well.
Right. So that's what we're doing right now. I mean, it's nothing obvious. So they're alive. They're fine. So we're doing more, like a kind of array of behavior and some other neurological testing. That's exactly the question, right? So the idea is what exactly, loss of 20%, the ability to respond, what that impact in the long term for a nervous system is ongoing.
Great. And the most recent question, Dr. Lanoila asked, do you have any insights yet into the regulation of immune cell therapy into the brain, immune cell entry into the brain through the blood-brain barrier, or is that too early to say?
Yeah, I think we're not really in too much about this. I'm aware of the several groups or study, like for example, post-venue region, or right now, like a very, very exciting area about the meninge place. I don't think normally there's in the brain parenchyma per se, in the capillary blood-brain barrier, there's so much natural immune cell entry. Maybe also in the circulatory organs, it could be. So I think there's several places, but not in the real in the normal situation. I think it's very hard to imagine in the majority of the brain parenchyma capillaries. But during disease or infection, it's hard to say.
Okay.
That's why I think we're interested in this type of question for the neuroimmune interactions.
Thank you. Wenlu Lee asks, the expression of the MFSD2A in endothelial cells is described in your studies. Would it differ in different regions of the brain? Is that known?
So it's a very specific. For example, I mentioned, in the brain, there's 7 places called the circulatory organs. They don't have blood-brain barrier, even though they have blood vessels. Those vessels are leaky for a good reason, right? So you can, sense all the hormones. You can do all the, So in those areas, MSH2A is not expressed at all. Choroid plexus not expressed at all. So I think in the brain areas that tightly correlated with the barrier function. So basically only in the normal barrier, functional barrier endothelial cells expressed there. In the 2 areas I mentioned, the barrier is completely open, MFC2A is not expressed there. Whether the slightly different level in hippocampus or basal, and that's very hard to, we don't know about that. But the things I mentioned is a day and night difference, basically.
Thank you. So I think we've made it through most of the questions that had already been posted in the chat. If anyone else has any questions, now is a good time.
Great.
Okay.
It's 10 o'clock, perfect timing.
So this has been a really amazing presentation. Thank you so much for your time and coming to speak with us.
Thank you. Thank you for step up in the last minute.
Of course.
Since Doc is there, I really hope you feel better.
He's listening in.
Yeah, I'll pray for you. Okay.
Okay. All right. So that's the end of Grand Rounds for today. Thank you all.
Thank you. Bye, everyone.